DNA sequences form genes, which code for proteins in all living organisms. Take the gene of one and put it into another, and you have a genetically modified organism, a GMO. For example, crops can be engineered to grow in dry, arid conditions by using the gene of a plant which grows in deserts. One of the problems people have with this kind of technology is the unknown long-term effects. What if this new gene is introduced into the wild where it does not belong? Can bacteria grow resistance to the same extreme heat? The truth is, we don't know. Scientists have been studying GMOs for many years. Research in support of genetic engineering is regularly coming out and points towards them being safe. Nonetheless, we have developed mechanisms of defense to prevent the DNA from getting out into the wild. Various barriers exist, which can be implemented in different ways. There's physical, chemical, and genetic barriers. Physical barriers include using well-enclosed spaces to prevent the GMO escaping into the wild. This is usually the case with bioreactors, large vessels used to grow bacteria in. The vessel is designed to not allow anything to leave. The obvious problem with physical barriers is that there's a size limit. Imagine trying to contain something as large as a crop field. This is why chemical barriers were created. Autotrophs are a popular example of this. The GMO is made dependent on a synthetic food source, food which is made in the lab, not commonly found in nature. If the food is not present, the organism dies. Finally, more novel genetic barriers are being developed by scientists. You know DNA and you might have heard of RNA, but XNA is a futuristic vision of biocontainment not currently available. The difference between all of these NAs, or nucleic acids, is that their chemical structure is slightly different from each other. They're essentially all written in a different language. XNA stands for xenonucleic acid, and is essentially a made-up language. The cell's machinery is reprogrammed to read XNA as opposed to DNA. This means that no other organisms will be able to understand it. As genetic engineering is increasingly implemented in agriculture, biotechnology, and clinic, it is important that the public becomes more aware of the mechanisms of defense being implemented.